My name is Jie Wang, MD, PhD. I'm a founder and also CEO of SymMed Medical.
My name is Howard Levin. I'm CEO and CMO of Deerfield Catalyst, but was one of the original inventors of renal denervation along with Jie, and have had a pleasure of working with him for many years.
Data from Street says once 2030, the market size could be as big as $2 or $3 billion. However, based on my own experience, because our device has been approved in China, start to getting the sale the beginning of December last year. Now we got sell about $1 million per month, more or less. And we can see the physicians are very actively to utilize the device to treat patients with hypertension, resistant hypertension, intolerant to the drug therapy, and patients want to reduce their drug burden. How do you think the RDN market in US?
I think that the renal denervation market in the US is going to continue to grow markedly, similar to the projections from the street. And I think that what is important is that people are now accepting the clinical data and adopting the use of renal denervation much more widely than they did previously.
Correct. Theoretically speaking, the best population for renal denervation treatment is a drug-resistant hypertension, which is about 10% of the total population of hypertension patients. But as you know, the patients with hypertension is a huge number. In China, more than 300 million people have hypertension. The 10% is huge. But in reality, it's not only this portion of the patients wanted to have device therapy, because without drug compliance issue, it has 24-hour, always-on therapeutic effects, and also with very stable blood pressure reduction effects. Therefore, patients who do not want to take drug therapy, they also want to have the therapy.
So let me ask you a question. You bring up a very important point about drugs. The study you did, the SMART study, was the only study that's ever been done that actually looked and quantitated and controlled for the drug use. And it was interesting to me to see that you were able to reduce the drug burden in these people while still meeting your goals. Could you comment on that?
Right. SMART studies have several features. Feature number one, it is the only study so far in the RDN field we can demonstrate RDN not only control blood pressure and also can reduce antihypertensive drug burden, because we realize if you do RDN clinical trial, there's no way, I want to say no way, to control patient antihypertensive drug taken, particularly in the sham group, because they went into the trial, their blood pressure is higher than 150 millimeter mercury office. Therefore, during 6 months trial, they will take drug. Once they take drug, their blood pressure went down. There's no differences between RDN and the sham group. In order to answer this question or deal with this question, and also answer very critical question in the RDN trial, because patients come to the office, once they use the device therapy, they will ask you 2 questions. Physicians have to answer that. Number one, doctor, how much my blood pressure can go down? Number two, can I take my drug away or reduce my drug? So SMART study answer those questions in one trial. Okay. Therefore, we use a combined clinical primary outcomes, control rate of office blood pressure, and drug burden. Therefore, during the study, we supply all the drugs. Once they get into the study, the physicians have to justify patients' drug therapy in order to reduce their blood pressure less than 140. But we have very restricted regime in order to increase the drug or reduce drug. And also ensure the drug adherence, we use a urine sample to do lab testing, see whether they compliant to the drug regime we prescribed to them.
So it's very interesting. You not only did what I thought was an extremely solid clinical trial, but you were able to show that there was something missing with the current methods of renal denervation, and that has to do with mapping. Could you explain to us what you found the difference between I guess we call it a hot spot, a cold spot, and a neutral spot?
Correct. If we look at the data from across the board, no matter it's a spiral study or the recurrent series study, and no matter they do main renal artery ablation, branch plus main artery, or recurrent ultrasound ablation on the main artery, the responder rate consistently between 60 or 65%. Okay, and also about 30% of the patients after the procedure, blood pressure did not go down. Some of them even went up. Okay, therefore, with this question, we come back to the basic science. We should look at the nerves around the renal artery, what they are, what components they have, and also what's the physiological functions they have. By now, the preclinical basic science study clearly showed there are parasympathetic nerves around the renal artery, not only sympathetic nerves.
Parasympathetic, really? I mean, most people believe that the kidney was only sympathetically innervated.
Right, that's from textbook. What we are doing today is break the textbook, rewrite the textbook. That's what we are doing today, including RDN. RDN is not in the textbook. Now we put RDN not only into the textbook and also into the guideline. Okay, yes, there are argues about whether it has parasympathetic nerves or not around the renal artery, but both sides accept one fact, which is around the renal artery, there are cholinergic neurons. There's no argue about this point. The argue is where is or what these cholinergic neurons belongs to. It belongs to parasympathetic nerve or belongs to parasympathetic nerve. The reason the study clearly showed those cholinergic neurons come from all the way down from the brain, or all the way up to the brain, the parasympathetic neuron, central neurons. They use whole brain 3D imaging, therefore use the virus which can across synapses. They can see those tracing appeared in the central parasympathetic neurons.
So is it correct to say that the cold spots that you find when you map relate to the locations of those parasympathetic or cholinergic parasympathetic nerves, and that if you ablate them, not only does blood pressure not go down, it actually goes up? The corollary to that I was wondering about, if you could comment on, it seems that irrelevant to whether it's RF or whether it's ultrasound, if you ablate those cold spots without knowing they're cold spots, you can actually be detrimental to your goal of reducing blood pressure. Is that correct?
Correct. The animal studies have demonstrated what you just said. In animal studies, investigators ablate the cold spot. After ablation of those cold spots, they do observe the increase in the blood pressure.
It seems that if you do, for example, ultrasound, and you're doing circumferential ablation all at once, you're going to get cold spots, hot spots, neutral spots,
Exactly.
no matter what. So
Exactly.
very interesting.
Why is 60 to 65% of the responder rate? Because the anatomy study already showed parasympathetic nerves around the renal artery is about 20%. Sympathetic nerve around 70 to 80%. Sensory about 8 to 10%. So without mapping, your probabilities is about 70, 60%.
That's very interesting. So in the absence of mapping, whether you use RF or ultrasound, the maximum benefit can only be achieved by doing the mapping, it seems, to not increase, have increases or, opposite direction effects of the cold spots.
Correct. Before we get a detail of those cold, hot, and the neutral spot, I want to briefly say how do we do the procedures. We have a catheter. It has dual functions. It can used as a stimulation catheter, and also it can be used as ablation catheter.
So you don't have to exchange catheters.
We do not need to exchange catheters. Therefore, we put the catheter tip all the way down to the distal of the renal main artery. And then we deliver electronic stimulation. If we see increase in the blood pressure, that spot identified as a hot spot. And then we ablate. After ablation, we deliver the same electronic stimulation again. If we still see increase in the blood pressure, meaning there is no, this ablation is not effective enough, so we deliver the second ablation.
So what you're saying is, right now, people are only basically ablating each spot once. They may ablate many spots, but they're only ablating each spot once.
Right.
And so you may get inadequate therapy if you don't know that that spot has not had technical success, so to speak.
Right. If it is not a hot spot, it either is harmful or it is unnecessary ablation. So by this procedure, we call stimulation, or call mapping, ablation, and confirmation. And then we treat the entire renal main artery from distal to proximal.
But doesn't that take a lot of time? I mean, when people think about mapping, they're always, thinking, oh, it takes so much time to map.
Okay, let's data speak. Okay, let's look at the data. Okay, first of all, if you do main artery plus branch ablations, you need to go to all the branches. So the data showed it's about 99 minutes. You can look at that from the data package to FDA. Okay, so because you need to go to all the branches.
That's for RF.
That's for RF,
simplicity.
For simplicity and for spiral. Okay, and if you look at the record, they only tell you the portion of the story. They say the ablation take 8 seconds. Correct. Once your catheter in the correct location, 8 seconds. But before you make your catheter in the correct location, you need to exchange the catheters. Shoot the contrast from proximal. If there's no contrast leak, you can ablate. Otherwise, you have to change another catheter. And before you use catheter, you also have to use Avas catheter to measure the size of the renal artery. They have 7 sizes. Therefore, the time is very comparable to what we're doing mapping selective denervation, because we only do renal main artery, and also the length of the main artery is limited. And also the SOP is you do each side 5 millimeter apart, 90-degree turnaround. Therefore, we only do about 4 or 5 hot spots per main renal artery. So time is about between 70 to 80 minutes. And you use much less contrast as well.
Now, I understand you have a Gen 2 device that can map the entire circumference at once. So you don't need to go 90 degrees. And by using it, you say you pick up more potential hotspots.
Right. We have two generations of our devices. Okay. Number one, where Jie Wang is a single electrode. Okay. From our clinical practice, real world, we found on the one cross section of the renal main artery, sometimes it has two hotspots. And also the current Jie Wang catheter system, the physician has to read the increase or decrease blood pressure, then make a decision where is hot, where is cold, and the neutral spot. The Gen 2, it has three electrodes on the same cross section of renal artery. Therefore, they will not miss any hot spot. And also it's computer driven. The computer will tell you where is the hot spot, where is the cold spot, where is the neutral spot. That will more time efficient and friendly use system. People argue, said, do we need a mapping? I think the question should be very straightforward. Okay, mapping or get feedback during a procedure is unmet clinical need. Okay, any therapy need treatment and a diagnostic combination. The perfect example is in the coronary stent, PCI. In the past, we only have stent. We do not have FFR. So the treatment endpoint on the table was anatomy judgment, which is the fluoro. Okay, see whether the stenosis has been dilated. But however, after FFR, that's a functional diagnostic assessment during PCI. The mapping for renal denervation is the same thing. You need a feedback during your procedure. Also, you need a judgment to see whether you have effective ablation or not, or you even have harmful ablation or not.
So, one, technical success. Two, identification of the spots that need to be ablated or should be skipped to optimize therapy. Is that correct?
Correct. Therefore, we are able to increase our responder rate to 80% from 60, 65. Interesting. I work with a Credia team together for more than 30 years. We learn a lot from Mark Galfin. The number one thing to do a startup company, SyMap is not anymore a startup company. It's already been 13 years. So we built up our IP, solid IP system from day one. Now we have more than 20 IP issued in US. We have more than 10 IP issued in EU countries and more than 10 in China. We also covered Canada, Australia, even Russia.
So that means that whatever geography you're going to work in, the mapping especially will be well protected.
Correct. We are not only protect the core idea or patent, mapping, ablation, confirmation, we also covered the device design, the stimulation parameters, the combination with a 3D system, and so on and so forth.
Renal denervation has potential opportunities not only, as Jie says, in hypertension, but in heart failure, chronic kidney disease. Some people have used it to reduce AFib burden. So I think that there's a number of different things that can be looked at in addition to hypertension. The SyMap's device gives us a huge opportunity to not only move forward with the treatment of hypertension, but to expand well beyond that into other areas.
I want to celebrate today because we really started the renal denervation together.
Long time ago.
Long time ago.
I think that it's important to note that the SyMap device can do the same ablations as any existing device, whether it be RF or ultrasound, but can do it much more effectively by not only identifying the areas that should be ablated, not only avoiding the areas that should not be ablated, but making sure that you have technical success, which has been the big missing part of the entire renal denervation program.